So today we are covering bacterial growth and control. Let's get into it. So what we're really talking about are kind of two aspects. So the second half of this talk is actually going to be talking more about disinfectants, different ways that we sterilize equipment, things like that. But the first part, we need to talk about some of the actual ways that bacteria and us too can actually control things, because it makes sense if you're going to, if you want to kill a bacteria with heat, well, then we should probably understand, what kind of temperature the bacteria actually normally like to live at. So we're going to talk about lots of environmental requirements for bacteria. So that's your temperature and your pH. Those are pretty straightforward, but we're going to talk about some other things like atmospheric pressure, how much salt is actually around. One of the big things that we do need to talk about first right off the bat is obligate versus tolerant. So I'm going to be talking about, what kind of temperature do bacteria like to live at, or what kind of salinity do they like in the liquid that surrounds them. And what's important is that we're usually talking about something we need to talk about is obligate versus tolerant. It's a really straightforward idea. Lots of different things are going to show up this way as we talk about these different requirements. So obligate means that you are obligated to exist at that specific place in order to reproduce. If you're tolerant, that means you can tolerate one of these aspects. So like if you're cryotolerant, then you can tolerate cold things. That means you can't reproduce, but you're okay with it. If you're obligate, like a cryo-obligate, then that means you like it really cold, and that's where you're obligated to live. So speaking of temperature, let's get into it. So temperature is usually the first one we talk about. So we've got this nice chart here. We've got our psychrophiles, is probably the more technical term for it, but I like cryophiles. So cryophiles, phile meaning love. You're going to hear that term a lot today. So phile just means loving. And so cryophile means cold loving. And so these are bacteria, or I'm going to say bacteria a lot, but it can be eukaryotes or archaea as well. But for the most part, we're usually thinking about bacteria, except when we're talking like our extreme organisms, the ones that can survive at really cold temperatures or really high temperatures, or extreme pHs or extreme alkalinities or things like that. But your cryophiles like it cold. Your mesophiles are what we typically think of. Most of your pathogens are mesophiles. They like it, 30 to 40 degrees. Usually 37 is a pretty safe number. Your thermophiles, those are the ones that like it a little toasty. And this is where we start getting into some of our archaea. We have a lot of extremophiles is what we call them, that like these extreme places because they've learned a way to actually survive in these places that you wouldn't normally survive in, which is good for them. Yeah, it's like really intense and extreme, and they have to sacrifice a lot of their life to be able to survive in these conditions, but they're not competing with other organisms. And so it's kind of that push-pull of what you actually need. So thermophiles, they like it pretty toasty. I mean, 45 to 70 degrees Celsius. I mean, that's getting up there. You can get into the hyperthermophilic range, and there's a few that actually exceed that, like extremothermophiles. But for the most part, you can't really get much higher because once you start hitting 150 degrees Celsius, that's when your hydrogen bonds break. And hydrogen bonds are what hold your proteins together and your DNA together. And so when you start heating up that hot, we have ways, and bacteria have ways of preventing that damage. But after a certain point, even the enzymes that protect the damage are going to start breaking apart. And so that's how you actually end up losing, the structure of your enzymes. And now you can't hold together and you die. So going from temperature to pH, remember pH is just how many hydrogens are floating around, basically. So if you have mostly hydrogens, not a lot of hydroxide ions floating around, then you have a low pH. If you have a lot more hydroxides and not very many just hydrogens, then you're more alkaline or basic. If you're floating around 7, that's pretty neutral. So 7 is our neutral one. So your hydroxide and your hydronium ions are in balance. If you like a more neutral pH, then that's going to put you as a neutrophile. That makes perfect sense. If you like it a little basic, we call them alkalophiles. There's also acidophiles. Like let's say a bacteria like Helicobacter pylori is what causes stomach ulcers, and it loves to live in the stomach, and it likes to dig through your stomach lining and your mucosal layer in your stomach and actually get in there and cause ulcers because that's a cool place to live apparently. And so we would call them acidophiles. Now there are a lot of bacteria that are acid tolerant. So I talked about obligate versus tolerant, right? So acid tolerant bacteria are like a pathogen. Let's say you ate a pathogen like salmonella, and it wants to get into your intestines to make you sick, or maybe like E. coli, like a a version of E. coli that can cause like diarrhea or something. And so it gets down into your stomach, but it has to survive that acid. A lot of them aren't necessarily loving being there, but they don't die. So they're acid tolerant. The bacteria that I worked on in grad school, which is lactobacillus, is acid tolerant. So it didn't like living in acid, but it actually, didn't die. And so it could actually get into the stomach, or down into the intestines, which was good. So going from pH to salinity, something we don't talk about too much, but halophile is the term that we use, salt-loving. So that's generally reserved for like 4 to 10%. So those are the bacteria that really like it. They're having some kind of requirement. There are certain types of bacteria, Staph aureus, which is one that you've probably heard of. It's halotolerant, and so we can actually grow it on media that will be slightly salty, and that prevents a lot of bacteria from actually surviving. So like an E. coli won't grow on media for Staph aureus, but Staph aureus does grow. And so you can have it even just slightly salty, and then you can just prevent the growth of some organisms, which is something that we'll talk about later. If you really want to keep organisms from growing on stuff, then for the most part, like salting your food or creating fermented foods, those are really good ways to keep organisms from growing on stuff. I mean, that's like the whole point of like jerky or salted meats, like salted pork from like, I don't know, the Middle Ages or something. Obligates require really high percentages. We have to think about like if they have a ton of salts around them, then they have to have a way to deal with that and still retain the water that's inside of them, because that's the whole problem. Is if you have a ton of salt outside of your cell membrane, then all the water inside wants to go back out, which is a great way to kill bacteria. But so if you're an obligate, then you kind of like it, and so you have to have ways to deal with that. So the actual killing of a bacteria using salinity, or the amount of salts around, it doesn't have to be sodium chloride, but the amount of salts that are around, that would be osmotic pressure, right? Like the movement of water out of a membrane. Barometric pressure is something else that we can talk about. So barophile or piezophiles, those are like pressure loving. They live deep down in the ocean. That's where you're going to see like down at those, the heat vents that are way down there, those volcanic vents that we've talked about before, where kind of extreme bacteria can survive and extreme organisms can live. Those would be barophiles that can survive really high atmospheric pressures. And so in this case, they're just looking at like the growth rates of different bacteria. And I mean, just like with salinity, I mean, some of them like it, they're tolerant. Some of them really like it a lot, and so those are barophiles. And then some really like pressure and have to have it, and those would be your extremophiles or your extreme barophiles. Piezo is another term for pressure. Piezoelectric is something that you may have heard of. That's a way of generating electricity using pressure. The best example that I can think of would be like if you installed piezoelectric walkways, then you could have people like as they're walking, they're generating electricity as they walk. Or you can install piezoelectric like exercise equipment, and so like every time you're exercising, you're transmitting some of that energy into an electrical system, and you're generating an electrical charge. That may be where you've heard of it. That's not exactly what it's talking about here. It's just talking about piezo, which means like pressure. So now let's move on to nutritional requirements. I mean, bacteria, basically like us, they're living creatures. They need tons of different nutrients. What is a nutrient? I mean, that's such a generic term. I don't like it a lot because it's so overused, but I mean, they need organic or inorganic chemicals. Like you have to build stuff. So you need to perform anabolism. You need to perform catabolism to get energy out, and just all kinds of compounds are needed for that. Mostly, like, they're all your basic ones, right? You need your carbon compounds, your oxygen, your nitrogen, your hydrogens. And then if you're going to make ATP, which everybody makes ATP, then you need phosphorus too. But everything else is for lots of other things. And so just it's not that crazy to assume that, you know, humans need micronutrients, and therefore bacteria probably need micronutrients for all kinds of random stuff too. Now, this brings us back to something we've talked about before, which is your terminal electron acceptor. I've spent so much time talking about this when we talked about metabolism, which was the lecture last week. So your terminal electron acceptor is that thing that's right at the end of your electron transport chain, right? It's the last thing that your electrons need to be put onto in the electron transport chain in order to keep putting NADHs and FADH2s in order to put those hydrogens on the other side of a membrane. And so you have to have your terminal electron acceptor. Oxygen is the one that aerobes use. Lots of creatures also can be anaerobes, so they don't use oxygen. Some are facultative anaerobes, so they can use oxygen or something else. Those are things we've talked about before, but we actually need to talk about, there are 5 actual categories that we can use to separate out our creatures. Usually we're talking about bacteria. So here is our list of 5 different ways to categorize bacteria. They will exist in one of these 5 places, okay? Humans, lots of bacteria are obligate aerobes. That means they can only use oxygen as their terminal electron acceptor. Now, we also know that there are facultative anaerobes. Those can use oxygen, or they could use something else. They can use an alternative terminal electron acceptor. It turns out there are these creatures called microaerophiles. Microaerophiles, they are aerophiles in that they are aerobes. Okay, so that if you follow that term, so the term aero is referring to oxygen usually. So if you're an aerophile, you like oxygen. And if you like a microaerophile, that means you like a small amount of oxygen. So usually that can't exceed 10% in whatever you're in. But you do want to use oxygen as your terminal electron acceptor, but oxygen can be kind of toxic at high concentrations, even for humans. And so you don't want to have too much of it there. And so a microaerophile is an obligate aerobe, but we can separate it out because it actually prefers to grow at lower oxygen concentrations. So then we hit facultative anaerobes, which I just talked about a second ago. Facultative anaerobes can use oxygen. Of course, they are not obligate aerobes, though. They are facultative anaerobes, so they can use an alternative terminal electron acceptor. There are two other exceptions that we talk about, okay? So they're both anaerobes. So an obligate anaerobe is the easier one to think about. So when we talk about anaerobes, not facultative anaerobes, but just regular old obligate anaerobes, they're obligate to be anaerobes. And so they like to use an alternative terminal electron acceptor. Oxygen is typically toxic to them, okay? So they can't be around oxygen, or at least the levels of oxygen need to be really low. Aerotolerant anaerobes don't use oxygen, but it's not toxic. So that's an important distinction. So if there is oxygen present, they just don't really care. If you're an obligate anaerobe and there is oxygen, you don't like that. You have to have just your alternative terminal electron acceptor. And so aerotolerance could basically grow anywhere. If you want to know what kind of organism you have, then one of the things that you can do is grow them in special media that doesn't allow oxygen to penetrate very far. And so it actually penetrates at like a specific rate. And so you would only have, like, your oxygen would be higher concentrations at the top of your media, and there would basically be no oxygen at the bottom. And so you would put your bacteria in here and you would let it grow, and then you see where it actually physically grows. It's kind of like turbid, or it's like cloudy where the bacteria are growing. And then you just look at it and you say, okay, well, let's look at where our bacteria are growing. Okay, these bacteria are only growing near the top. Well, that must mean they like oxygen, right? So because oxygen's only near the top, it's not near the bottom. Okay, so this must be some kind of aerobe, right? It's using oxygen. Now, number 1 looks very similar to number 4, but you'll notice here they must be using oxygen because they're growing near the top, right? But they're not growing, they don't seem to like right at the top. They like it down here a little bit. Well, that must be our microaerophile, right? Because microaerophiles do like to use oxygen as their terminal electron acceptor, but they like it micro level. And so the amount of oxygen decreases as we go farther down, and so therefore this must be a microaerophile. So jumping off of that makes this one pretty straightforward. I mean, there's not supposed to be any oxygen down here, and these guys can't grow anywhere up here, so they must not like oxygen. Well, that must be our obligate anaerobe. So they're using something else to actually perform their electron transport chain here, and so these would be your obligate anaerobes. So that leaves two things left. That leaves our aerotolerant organisms and our facultative anaerobes. And we know that facultative anaerobes like to use oxygen if they can, but they can use something else. And so if we look here right in the middle, we see right at the top where there's lots of oxygen, we actually see a larger concentration of our bacteria. And then we can see them interspersed throughout here. And so they must be able to use some kind of alternative terminal electron acceptor because there's no oxygen down here and they're still growing. But they are kind of concentrating at the top. So this would be your facultative anaerobe. And you can see the big difference here is they're not congregating at the top. So therefore, there must not be an advantage to using oxygen. Because remember, oxygen is the best terminal electron acceptor. And so if you don't need to use it, I mean, it is the best, but like if you can't use it, then what's the point? Well, being aerotolerant is fine because, I don't know, you can still live here and exist in this space and use up the nutrients that are there other than oxygen, but it's not toxic, unlike over here. Now, these cells are all growing at different oxygen concentrations, and so what we need to do is talk about cell growth. Now, lots of organisms, basically every kind of biological organism, including humans, follows this exact same curve, at least on a population level. And so we have 4 different phases. So on the left is our number of cells, and on the bottom is over time. You always start out with a lag phase. So the lag phase is where you're just kind of getting ready. You have some bacteria, they're starting to grow, but it takes a little bit of time to build up that momentum into your log phase or your growth phase or your exponential phase. And that's log growth. So it's growing so fast and you're undergoing so much binary fission, which we'll talk about in a little bit. It's just this rapid growth phase. But then eventually you start running out of the nutrients or whatever rate limiting reagent you're dealing with. So that could be your food source, that could be your oxygen source, that could be your electron source. Who knows? It doesn't really matter, but you're starting to use those things up because you've grown so fast. As you start using those things up, then you eventually, you're just kind of plateauing. So the rate of bacterial death, if we're talking bacteria, is the same as the rate of bacterial growth. And so you enter the stationary phase. But eventually, you're going to start using those things up. Those nutrients are going to be gone, like oxygen or sugar or whatever. And then you start to die. And then the rate of death exceeds the rate of growth. And then you enter your death phase. So that's bacterial growth. Now, what's really interesting is when bacteria start hanging out in large clusters, which is what you're going to get as you exit your lag phase, enter your log phase, and the early stationary phase too, you can get these huge bacterial colonies. Those are what we typically grow like on agar in the lab. And something that bacteria can do is they can take all of their glycocalyces, so that's their slime layers, or their capsules, but usually we're talking about slime layers, and they take their glycocalyx and they can use that to actually control their environment. We call that environmental control biofilms. So a biofilm would be a collection of colonies of all the glycocalyces, all these bacteria. It can be one species of bacteria. It could be multiple species of bacteria. But they're these massive bacterial colonies, and they start acting like larger scale organisms. And so in this case, on the right here, you can see this is an example of like a Staphylococcus bacteria that's growing. This actually came from a paper that talked about if you get a staph infection on like an artificial limb, or let's say you got your hip replaced and you have a fake hip, you have like a metal hip in there, then a lot of times they can get bacteria in there and they can form biofilms, and they can be really, really dangerous because they'll start growing on the bone and they can start eating away at the bone, or they can just start infecting that socket that the artificial bone is in, and it can start to cause a lot of damage. You can see here, it's really interesting. We usually think about bacteria as functioning by themselves, but what can actually happen is the bacteria that are closer down to the bottom, your teeth are another good example. So like a bacteria that's really close to your tooth, let's assume that this biomaterial here is your tooth or the substratum, then if that's your tooth, then the bacteria that are really close to that are going to be digesting your enamel. They're going to be probably undergoing anaerobic respiration because the amount of oxygen there is going to be really low. But then as they start to grow, they kind of pile on top of each other, and they can start getting to the top here, and those bacteria can actually be released to go form new biofilms. But why do they know to do that? Well, that's a really good question. We call that quorum sensing. When bacteria and other cells too are sort of talking to each other, and they can send different signals to each other. And it gets really creepy because bacteria, we think about them living by themselves and being like these individuals, but they actually function together as like a larger scale organism and can do a lot of damage. Biofilms are a great way for a bacteria and a bacterial colony to control their environment. Now, we can artificially control bacterial environments, and we call those growth media. So growth media would be like a way for us to artificially grow bacteria. It's actually really hard to grow a lot of types of bacteria. We know that there's tons and tons of bacteria that live on this planet, and archaea as well, that need nutrients that we just can't provide them in typical agars or broths. So agar being solids and broths being liquids. Basically, your most basic growth media is essentially just like chicken soup or like a vegetable soup. That's what like a generic growth media would be. So like you can have a general media, and I'm glossing over a lot of different types of media. There's lots of sub-medias, but like a general media would be just, I don't know, like some kind of broth that you can grow bacteria in. A selective media would be something that can grow a specific type of bacteria. And so if I only want to grow gram-negative bacteria, or I was talking earlier about Staphylococcus bacteria being halotolerant, well, that would be a selective media where I can just put some slight salts into that media and then it selects just for Staphylococcus. A differential media would be something that's going to tell you something about the bacteria, which of course a selective media could also tell you something, but differential is usually specifically for areas of metabolism. And so on the top here, we've got like our general nutrient media. There's some E. coli and some Staph aureus growing on here, and that'll make you sick. And then over here, we switch to McConkey. So McConkey has an indicator in it, a pH indicator. And so if you have E. coli on there, E. coli are really interesting. So they can undergo not only oxidative phosphorylation, so they're using their terminal electron acceptor, which I think is oxygen in this case, because they're not under anaerobic conditions, and then you have your E. coli, but they can also undergo fermentation. So sometimes they may want to use their fermentation pathways to generate whatever byproduct that is, which they can use to do things. And so in this case, they have a pH indicator, and so you can see it's getting really dark purple here. Staph aureus doesn't even grow, but you can put Salmonella enterica on here, and you can see they look different. And so it's allowing you to differentiate between these two kinds of bacteria. Another really common one would be blood agar. So that's where you basically take like a generic nutrient media, like, I don't know, TSA, and you put blood in there. Usually it's sheep's blood. If you make it, it smells horrible. So usually we just buy it from like Carolina Biological or something. And then down here, you put your bacteria on here, and different bacteria will engage with red blood cells in different ways. Basically, can they destroy the red blood cell and eat it or not? And so we call that hemolysis or hemolysis. And hemo meaning like blood, and lysis meaning to cut or to break. And so if they can do that, then we call it one of different classifications. Gamma is actually no hemolysis. I don't know why they're just like non-hemolytic. They call them gamma hemolytic. It's so dumb. But so staph epidermidis is like a really generic one, but certain types of E. coli and certain streptococcus, so strep pyogenes will cause lots of disease. And you can see they're destroying all the red blood cells. The agar is supposed to look like this, but they're killing this red blood cell. But that's differential. It's telling you, okay, like I could differentiate, if the bacteria does this, that differentiates it from bacteria that don't do that. So speaking of bacterial growth, we just need to talk about how bacteria actually do grow. The normal way that they do it, at least the most typical way, would be binary fission, which is like mitosis. Basically, you make all the things that you need to make two bacteria, and then you split down the middle with your little septum there, and you separate. Most E. coli take about 20 minutes. Some bacteria take 30 minutes. Some bacteria take a really long time. So like tuberculosis is known to take like days to double, and it's a pain in the butt if you're trying to grow it. But E. coli grow really fast, and so you can have something go in like 20 minutes, you're going to double, and so you can go from one bacteria to millions of bacteria in like a day. So that should make you really uncomfortable. That's why you don't leave food out at the wrong temperature. Some bacteria can do budding. Budding is more understood in the terms of like yeast. So they basically have like this little tumor looking thing grow off, and that can separate and become a new bacteria, as opposed to basically making one megabacteria and splitting it in half. For the most part, we're talking about binary fission. How we actually count bacteria is a great question. There are tons of different ways to count bacteria. Probably the two most common ones are if you have a lot of bacteria and you want to know how many are in there, you need to dilute it out. So let's say in your original solution, you have like 1 million bacteria per mil, or per milliliter. So if I have 1 million of bacteria per milliliter, that's a lot. So I need to dilute that down because there's way too many to actually count. I can't take 1 milliliter of this and put it on a plate and expect to be able to count 1 million colonies. That would be absolutely ludicrous. And so you start diluting it down. So I do like a series of 1 to 10 dilutions. That's called a serial dilution. And so if I do a 1 to 10 dilution, then if I had 1 million per mil here, now I have 100,000 in this one. And now I have 10,000 in this one. And now I have 1,000 in this one, and now I have 100 in this one. And so you just keep doing that, and then eventually you get to the point where, let's say you had it down to, you have 10 per milliliter. Well, if I take 1 milliliter and put it on a plate, then I grow 10 colonies. I can count 10, and then you just go backwards. You're like, okay, well, I did a 1 to 10,000 dilution, and I ended up with 10 colonies. So I multiply it 10 times 10,000 is 100,000. So I have 100,000 bacteria per mil. Pretty straightforward. Now, that's if you have a lot of bacteria. This is what you would typically do in a lab situation where you're trying to grow a lot of bacteria because, I don't know, we want to grow a lot of them and see what they're doing. Another way that you might want to count bacteria would be in terms of like, let's say you're working on a water treatment plant where you have, your volumes are massive. So you've got hundreds or millions of gallons of water, or like a pool, and you want to know how many bacteria are in the pool. Well, there's a ton of water, which is what's diluting out your bacteria. So you're already super diluted. And so one of the things that you can do is you can say, all right, I'm going to put this membrane here and I'm going to run 100 gallons through it. And then I'm going to grow any bacteria that fell through that. And so you could say, okay, I got, let's guess here, let's say there's 100 colonies here. And so you could say, all right, there were 100 colonies that grew in 100 gallons. So that means I have 1 bacteria per gallon. And that kind of makes sense, right? Something that we talk about, it's a term that gets thrown around. It's called colony forming units. There will be a chart later that shows that term. Basically, you can't guarantee that a bacteria is not undergoing binary fission and just hasn't split yet, or that two bacteria just aren't really lovey-dovey and just stuck together. So you can't prove that a single bacteria is starting a single colony. That's just sort of physically impossible to prove, but we can kind of assume it. And so if we're making that assumption, instead of saying, okay, I have one bacteria per gallon, well, what I would say is I can get one colony forming unit per gallon. But in all practical purposes, it just means one bacteria per gallon. That's basically what you're saying. So now we need to move on and start talking about the fun stuff. We're going to talk about how to kill bacteria. So I've got this chart here in the PowerPoint. You can look at this. You can pause this video if you really want to. But we're going to go through most of these methods of actually controlling bacteria physically. And then we're going to talk about chemical methods. And we're going to talk mostly about the liquids that we can use. And so let's get into these physical methods. So there are some terms that we need to talk about first because they're going to get thrown around a lot. I don't like being very term heavy, but these are just important ones that we need to understand. I really like this chart too. You can see it's using the same terms. So sterilization means you've completely removed everything. So when somebody says, oh, this thing was sterilized, that has a specific meaning. That means there's absolutely nothing living there, including endospores. You could argue including the exclusion of prions as well, but prions do require special levels of sterilization. Luckily, we don't really deal with prions too often. We will talk more about prions when we talk about viruses and prion diseases. That's like mad cow disease. It's an infectious protein. Yes, that's really weird, but basically endospores are the gold standard. So if you're trying to make sure that you have completely removed everything, you throw some endospores in a special container, and if those die, then you know that absolutely nothing else has lived, because we know that endospores are almost indestructible. Disinfection is like your next step down. So sterilization, there's nothing living. Disinfection would be like the removal or destruction of pretty much everything, but it's not guaranteed to get rid of endospores, which endospores are a huge problem, especially in hospital settings. And so you need to make sure, like, what am I actually doing? Am I killing the endospores or not? If I'm just disinfecting, that might be good enough, but it depends on what you're doing. Antisepsis would be like on a person or a patient or something. So if you're trying to clean the bacteria, usually we say vegetative bacteria. So that's basically everything that's not an endospore. Vegetative meaning like functioning. Then antisepsis would be removing those things from living tissue, at least on the surface of a living tissue. So that's like an antiseptic. Chemotherapy is when we're actually getting it from inside the host tissues, when we're killing stuff. And chemotherapy is, I mean, that can include chemicals, but that could also include like antibiotics, which we're going to talk about next time. But if you're using a chemotherapy, it means you're inside of a tissue. So, I mean, that's usually like it's either ingested or injected or something like that.
Right, and then I see the disinfectant where it knocks it out in a minute, 1 minute. And is there a way we can do something like that by injection inside or almost a cleaning? Because you see it gets on the lungs and it does a tremendous number of the lungs. So it'd be interesting to check that. So that you're going to have to use medical doctors with. But it sounds interesting to me.
A really important set of concepts to get across is the effectiveness of treatment. This is one of those, it feels kind of like a duh factor, but it's actually really, really important to understand how we know how effective a treatment is, because that's going to decide what kind of like chemical or what method of sterilization you're going to actually use. I mean, if there's a ton of bacteria sitting around somewhere on like a chair, you're going to do something different than if there was a little bit of bacteria on someone's face. I like to think about the idea of someone pooping like on a hospital bed and there's like poop everywhere. So what are you going to do? Well, there's a lot of bacteria. The location is awkward. There's a lot of inorganic matter floating around. There's a lot of organic matter. And so like if somebody has diarrhea all over, well, you're going to deal with that differently than if someone has like your kid grabs a chunk of mud and throws it or something and they've got muddy hands. Well, that's a different issue. So the amount of actual organic and inorganic matter is very, very different. Other physical things too, like what's the pH of whatever you're trying to deal with? What's the actual temperature? I mean, it's harder to clean really cold stuff than it is to clean hot stuff, because temperature is just the vibration of molecules, and so like if you have a high temperature, it's generally easier to get things to work. That's why washing your hands with warm water and soap is typically better, because you'll find that it lathers up easier as opposed to like a cold temperature. How long you actually leave whatever it is you're on there is important. What are you trying to kill? If you're trying to kill endospores, then that's a really important issue. If you're trying to kill Staphylococcus aureus, maybe that you need to use a different kind of a disinfectant. If you're trying to sterilize something, maybe you need to make sure that it's not a heat-tolerant bacteria. If whatever you're trying to get rid of has a biofilm, that's really important. Biofilms are this giant, sugary, proteiny layer. Like, that could really affect what you're trying to do. And what's your disinfectant? Like, what are you actually using? You know, like if you're using bleach at a 2% versus bleach at a 5%, that could make a big difference. And so you can see on this chart, this is chlorhexidine, which we're going to talk about in a little bit as a specific disinfectant that we can use. But you can see these are all different types of bacteria and how long you need to use chlorhexidine at 4,000 milligrams per mil to actually kill them. And you can see the bacterial innate organism resistance is different based on the different bacteria. This next chart was actually on the wall at an HIV lab that I worked at the University of Pennsylvania, and it's talking about HIV. And so in this case, they took either cell-free virus, so that's virus that's just, just like floating around in whatever liquid they have, versus cell-associated virus. So cell-associated virus is basically you're taking the virus factory, and because they're infected cells, and those are like the virus is inside of a cell, and so it's almost like the virus has this huge level of protection because it's inside the cell, and so it's able to resist a lot more stuff because it's like a giant eukaryotic cell versus just a free virus, which is very, very vulnerable. And so they said, all right, well, let's take a bunch of this random stuff and we'll expose these viruses either with a cell or without a cell, and then we'll see after 1 minute what actually survives. And then we're trying to test, like, okay, if you're in an HIV lab, like, what do you actually need to use to sterilize equipment? Because HIV is really scary. And so they found here, like, a plus means that it actually grew within 28 days of being placed on cell culture, which is kind of cool. And so you can see here, vinegar, soap, bleach, those are all really good. 70% ethanol is actually typically pretty good, but this is like a ton. This would be like a whole vial of blood, 10 to the 6, like hardcore infected blood. It doesn't get through those cells. They're actually able to survive. And you can see that with lots of these ethanols. What doesn't work? Water for the most part. They tried weird stuff. Beer? That's strange. So when you get to those low concentrations of ethanol, basically that's not super helpful. But we're going to talk about ethanol later. So let's talk about how to actually kill things, and we're going to cruise through these pretty quickly. So what we're going to start out talking about, we're going to talk about how we physically kill stuff, so that's like your heat, radiation, then we're going to talk about mechanical removal, and then we'll move on to chemical agents. We're not going to talk biological agents in this section. We're going to save that for next time when we talk more about like antibiotics. So let's get into it. We're going to start with heat. So dry heat would be really straightforward. That's your Bunsen burners, your flames. You could also use like a benchtop incinerator, which we don't use in our lab, but I have seen those before. You basically take your sterile loop and you stick it inside like a super hot heating element. An important idea to get across when you're dealing with heat is your thermal death point. So that's the lowest temperature to sterilize a pure culture in 10 minutes. Okay, so the thermal death point is really important if you're trying to kill something specific. Like, let's say you have a culture of 1 million E. coli. Okay, well, if I have a culture of 1 million E. coli, what temperature do I have to keep that at for 10 minutes to make sure that they're all dead? That's a really good question. It depends on the bacteria. Your thermal death point is how much time you need to sterilize a pure culture at a set temperature. These are all just things that you would measure to make sure that you are maintaining a standard, because you have to have standards, especially for the sterilization of specific equipment, because, I don't know, you don't want people to die. So if that's dry heat, then we're moving on to moist heat. And so that's typically using steam to do something. So if you're going to steam sterilize something, we usually talk about it in terms of autoclaves. So right here is an example of an autoclave. We have one in our lab. We use them all the time. If you get into like a science lab, you use autoclaves to... They're basically pressure cookers, and so you send a ton of steam and you put it under pressure, and it forces whatever you're dealing with to get really hot really fast, and it kills everything. And so that's a way to sterilize something. The way that we generally test it is you would use test strips to make sure that there was steam nearby, which steam has to be really hot in order to just be steam, and you have to use endospores, and so you would have endospores in a little indicator, and then you would make sure that they died as well, because we want to make sure that there's pure sterilization. Sterilization is the elimination of endospores. Pasteurization, named after Louis Pasteur, is basically just heating stuff up. Usually we think about it in terms of milk, but you can pasteurize other products. And you're going to pasteurize it, you're going to heat it up so that you're killing most of the bacteria. You're not eliminating endospores necessarily, and you're not necessarily eliminating all the bacteria, but for the most part, it's disinfected. So it's pretty good. It's about as good as you want it to be. You don't want it to be too hot because then you're going to start degrading all the stuff that you actually want in there. So to kind of just talk about how important sterilization is and the use of an autoclave and actually cleaning things, I wanted to show this video of a sterile processing technician and basically the bowels of a hospital and what they do there to make sure that all of the equipment for surgeries is sterilized. It's a really interesting job that you can get trained for and make a good living at it, and you're doing a lot of cool stuff.
One of the most important areas of Mary Greeley Medical Center is right here. We're in the Sterile Processing Department, and it's where all the surgical instrumentation gets cleaned and sterilized for any upcoming surgeries. And not many people get to see this area, but I'm going to give you an inside look into the sterilization process. Each day, we see hundreds of different types of instruments that require hundreds of different ways to clean them, and each sterile processing tech must learn these to ensure that the instruments used are safe for the next patient. Instrument reprocessing begins in the decontamination room. This process begins by soaking and manually cleaning each instrument in a sink filled with a water and detergent mixture. This helps break down and remove the blood and other materials that can be left from surgery. We call this material bioburden. We use a variety of different types of brushes, syringes, and automated irrigators to remove the remaining bioburden from the instruments. After all the bioburden has been removed from the instruments, we rinse them and move them to the next step in the process, ultrasonic cleaning. With ultrasonic cleaning, we are removing the remaining biofilm that cannot be seen. Ultrasonic cleaning works by creating ultrasonic waves that travel through the water. These waves create tiny unstable bubbles, and when these bubbles come in contact with the instrument, they implode and remove the remaining biofilm. After all the biofilm has been removed, the instruments are rinsed and prepared for the final step of the decontamination process, mechanical cleaning. Mechanical cleaning is done in a washer disinfector similar to a high-powered dishwasher. This step removes anything that might be left on the instruments. The washer rack has spray arms that spin and clean the instruments using detergents and high-pressured spraying water. Each cycle provides a thermal disinfection so the instruments are safe to handle. When the cycle is complete, we can now inspect and assemble the instruments in preparation for sterilization. Assembly begins by inspecting each instrument to ensure that they are working appropriately. We then arrange these instruments into sets that will be used during specific types of surgical procedures. Each set has many types of instruments in common, as well as instruments that are unique to a specific surgical procedure. So each sterile processing tech is responsible for learning each instrument and how it works. This provides the surgical staff everything needed to provide excellent care for our patients. We then prepare the instrument set for sterilization by placing all the instruments in a mesh basket. The basket is then placed in a specialized container that is designed for sterilization. Some containers have a sealed hard outer casing, and some must be wrapped with a specialized material called blue wrap. Both types of containers are designed to allow the instruments to be sterilized in the container, and once sterilized, maintain the safety of the instruments while in storage. Sterilization is done by using different methods such as steam, vaporized hydrogen peroxide, and a variety of other methods, but the most common instruments that are used in a surgery are sterilized using steam. To make sure the sterilization process has been effective, we use chemical and biological indicators. Chemical indicators are placed on the outside of blue wrap or a rigid container and allow us to know at a glance that the instruments have gone through the sterilization process. They are also placed inside the container with the instruments so the surgical staff knows the instruments were sterilized and safe for use on our patients. The biological indicators help us verify that our sterilization process is effective enough to kill all the germs that may be on our instruments. This process helps us know we can provide patients with the best possible care. Once the instrument set has been sterilized, they go into our sterile storage area until they are needed for the next surgical procedure. This area is also the staging area for the next day's surgical procedures as well as potential emergency surgeries. Once the instruments are ready, we assemble them on a case cart to be sent out for surgery. Be sure to check out mgmc.org for more information.
So that was a video about sterile processing in a hospital. It's in Ames, Iowa. Very cool stuff. So now let's talk about, if we're talking about heat, let's talk about the cold. So we all know if you refrigerate something, it's basically slowing down the metabolism of the microbes that are growing. And so you tend not to, get mold or whatever. It can grow eventually, so things can spoil in the fridge. I mean, we've all seen that. But it does happen more slowly because chemical reactions happen more slowly. If you freeze something, so this is an interesting set of pictures that I found. This we have intracellular ice crystals. This is yeast, so it's not a bacteria, but we have intracellular versus extracellular freezing of a yeast. So this is like if you took a yeast cell and you just froze it, and you can see all these crystals. Everything inside is basically destroyed, especially the plasma membranes. What's really happening is some of the water that's actually inside of the cell is freezing, and we know that water expands when it becomes ice, and so that can form crystals that can be really, really damaging, especially to plant cell walls. A great example of this would be like if you have a garden and you have that first frost and you go out and look at your plants and they're all droopy and stuff. Well, what happened was the water that was in those plant cells has expanded and shattered those cell walls. And so you can see that happen. Basically here we have a yeast cell that we froze and all of its internal organelles, all those plasma membranes have frozen. You can also cause damage to cells by freezing the outside. And so if you froze the outside, you can destroy the outer cell wall or the outer plasma membrane at least, and that will also kill the bacteria or the yeast or whatever you're trying to kill. Using freezing to kill things is not a great idea. It's more of a better way to just kind of preserve stuff. But speaking of preservation, you could create, osmotic pressure, as we talked about with halophiles. So if I have a lot of salt around something, like I'm going to create like salted meat or like a preserve. The reason we call them preserves is there's so much of some kind of solute there, so like either a salt or a sugar or whatever, that most bacteria, maybe funguses have a little better time with it because they're eukaryotes, but for the most part, lots of things don't like to live there. That's why you can kind of leave honey just sitting out because there's so much sugar there that most things can't actually even consume it. Lyophilization I stuck on here, so that would be freeze drying something, and the lack of water can also be really good because most organisms need water to survive, and so if you completely desiccate something, so think jerky, then things can't live. Radiation is another way to actually take care of stuff. So ionizing versus non-ionizing radiation. Non-ionizing, so that's UV light. So we've probably all seen things like using UV light to sterilize things, to sterilize equipment. Basically, you're mutating DNA. When we talk microbial genetics, we'll talk a little bit about this. We're also going to spend some time talking about mutations and mutation repair. But when you use UV light, you're forming what we call thymine dimers or pyrimidine dimers, but usually it's thymine that we're talking about. And you can see here, if you're trying to replicate your DNA, well, these two thymines are stuck together. That's not going to be good. So basically, you're going to kill stuff. And it's very difficult for bacteria or anything, including us, to become resistant to this. So it's a really nice thing to use as opposed to a chemical that bacteria can become resistant to. Ionizing radiation is radiation that's super highly energetic, so highly energetic that it can basically generate ions. So ions have a charge, right? And so what you can do is basically force electrons to get so excited that they leave an atom, and then that's going to leave some kind of charge, and then that thing can then react. And so we see that with X-rays and gamma rays because those things, as the lower your wavelength goes, or the smaller your wavelength, the more high energy it gets. So there you've got your X-rays and your gamma rays, and there's so much energy in there that they can really excite atoms, especially in DNA. That's usually where we can see this. So you can get single-stranded breaks in DNA, you can get double-stranded breaks in DNA, and those are really, really powerful. So I'm going to show a video right now that shows a sterilizing robot. So this is going to use UV light. You may encounter one of these if you work in a hospital. When my wife was having a baby, we had just had a baby, and I was walking around because I had been up for like 24 hours straight, and I had no idea what was going on. And I'm just like wandering the halls of the hospital, and I saw these flashing lights. I was like, what is that? And I was looking, and I saw one of these machines. And so what one of these machines does is, and I'm showing the video now, it should show this inner bulb. It's a giant UV bulb that comes out, and it shoots a ton of UV light into a room in pulses. And as I said, UV light is really hard to resist, and it's a great way to sterilize a room. So that's our physical methods of control. We can also have mechanical methods, which I guess you could argue is physical as well, but that's basically filtration. And so if you're going to use a HEPA filter, which is a high-efficiency particulate air filter, it removes really small stuff through a variety of processes. If it's a really big particle, it gets stuck. If it's a small particle, then it can actually be attracted to the charge. Wibbly-wobbly, timey-wimey, sticky stuff that filters are made of. Other membrane filters, so I used to use a filter like this to strain out, this would be the media that I was trying to grow like cells in, and you need to make sure there's no bacteria if you want to grow eukaryotic cells because they would take over. And so you would put them through a nice little membrane filter here, and those are really good at removing really small bacteria. So now we need to move on to chemical methods of control, and these are the fun things. So here's a nice chart here. A really important concept to get across is that some bacteria are much more susceptible than others to different types of chemical control. And so you can see on this chart, which is a really, really nice one, it's pretty comprehensive. And basically at the top, we've got our things that can be killed more easily. And then as we go down, these are things that are not as easily killed. And so you can see we've got our endospores here, our prions are way down here. And so those are really tough to kill. In fact, none of these chemicals can actually kill prions. Prions usually need to be killed with either a hardcore autoclave, so like that's not a technical term, but a really powerful autoclave that's stronger than a typical autoclave, or incineration, because it turns out if you're just burning stuff, it gets rid of them. But for the most part, you can use disinfectants that run the whole gamut, and you can eliminate most of what you're trying to get rid of. But you can see your basic gram negatives and gram positives, okay, they kind of exist up here. But different organisms, as you go down, can resist things, but they kind of make sense. Like, so your spores, okay, yeah, awesome. Your tuberculosis, it turns out your mycobacteria, which they're saying acid fast, that's a specific type of stain to look for mycobacteria, they're much more resistant because of their cell walls to most disinfectants. And so we classify disinfectants, okay, if you can kill tuberculosis, then it's a really good disinfectant, and it's pretty much guaranteed to kill everything else on the bottom of this list, or at the top of this list, if you will. And so being able to just kind of generally rank bacteria is really a good thing to be able to know. So I'm not going to go through every single one of these, but I'm going to hit most of these. So for the most part, the way we're destroying things with chemicals is through either the plasma membranes being destroyed, or your proteins, or your DNA, your RNA. You're destroying these things usually by oxidation. So for the most part, you're trying to either disrupt a plasma membrane, or you're trying to oxidize something by ripping the electrons away. So the first one we're going to talk about is alcohols. So that's your ethanols, your isopropanols, which is like rubbing alcohol. They denature proteins. They can dissolve the lipid bilayer. You do have to have the presence of some water, so that's why you have to have usually minimum 60 to 90% ethanol is generally what you would use. When I worked in research labs, we tended to use 70% ethanol. Most hand sanitizers are around 60%. We can see here, if you have pure ethanol, bacteria will grow, G meaning grow. If you put a little bit of water in there, it allows it to pass through that plasma membrane and can really kill bacteria relatively non-specifically. It's very tough for bacteria to resist ethanol degradation. Aldehydes is another example. So you've probably heard of formaldehyde. That's how we typically embalm bodies. We use formalin, which is just a polymer of formaldehyde. So formaldehyde by itself is extremely reactive and is very tough to maintain. And so we use a polymer of it that can just survive longer. But they cross-link functional groups. And so as opposed to degrading things, basically what aldehydes do is they sort of freeze them in place, which is why we use them, to embalm bodies. Formaldehyde is a great example of something that you could use as a liquid, but you can also aerosolize it. There are situations where you would want to aerosolize a disinfectant to be able to make sure that it's getting in all those little nooks and crannies. And so if you're doing that, you need to use a chemical compound that can become aerosolized very easily. And so methanol or formaldehyde is really easy to do because it's a really small molecule, so you can turn it into a gas relatively easy. Biquinides is another chemical that shows up sometimes. So chlorhexidine, which I showed earlier, that was this little chart here. So that was chlorhexidine. Really, really interesting set of chemicals. They have to have at least one of these biquinide structures right here. And so I added these hydrogens here. And so what happens is under acidic conditions, which most biological systems are acidic conditions, then these nitrogens right here can become extra protonated. So they have this extra hydrogen here, and nitrogen only normally wants 3 bonds. And so if I throw another hydrogen on there, it now has 4 bonds, which gives it a positive charge, which will attract this molecule towards the bacterial cell wall, which bacterial cell walls are generally negative. And so it can attract this thing in there and it can help destroy those cell walls. Another set of chemicals would be your halogen-releasing ones. I mean, that's just your sodium hypochlorite, which is bleach. There are other types of hypochlorites. But basically, you've got this oxygen right here, and oxygen normally wants 2 bonds, right? And so you can see it only has 1 right here, so that oxygen is pretty upset about that, and so therefore really wants another bond. It really wants to pull electrons from somewhere. Well, if it's doing that, that means it's going to oxidize something. This oxygen can become reduced, thus oxidizing something, and we know oxidation is like the breaking of bonds. And so when you do that, you're able to have a powerful oxidizing reagent, and so it's going to wipe out things hardcore, and so that's why bleach is so powerful. Iodine-based compounds you may have heard of. If you use them on like a surface, those are the things that kind of look yellow. So you may have used betadine before. I had to show this ad for betadine because I encountered it as I was building this presentation, and it's absolutely insane.
Betadine. Fear not, my child. Betadine taps into the elemental power of iodine to shred germs. It is truly the bane of bacteria.
Betadine. Unleash the power of Earth's antiseptic.
So yeah, somebody paid a lot of money to have that be made. They're clearly targeting like Gen Xers and millennials that watched He-Man, I think. I don't know. So moving on to heavy metals. So those are like any kind of heavy metal that's actually on the periodic table, but the ones that we generally think about that are antimicrobial would be like your silvers and your mercuries and your coppers. We see those in lots of different things. Copper sulfate is an algicide or whatever. Basically what they're doing is they're able to find thiol groups, which is sulfurs, or amine groups, which that's in like your DNA and your proteins, and they can denature proteins. And so your thiols and your amine groups are what are going to help have secondary and tertiary structure for your proteins. And so basically these heavy metals will go in and break up those bonds. A lot of heavy metals are also toxic to humans for that exact same reason, so that's why you generally don't want mercury poisoning. Phenols, the most common one that you may have heard of is triclosan is a really common phenol. It's a bisphenol. You see you got 2 of these phenol groups here. Triclosan used to be in what we would call antibacterial soap. So that was the active ingredient in antimicrobial soap. And the FDA banned triclosan because it was causing antimicrobial resistance, not necessarily just against phenols, which it was, but what you would do is when a bacteria was becoming exposed to phenols, or at least triclosan, it would induce the production of efflux pumps, which is something we'll talk about with antibiotics. But an efflux pump is one of the ways the bacteria can resist antibiotics. And if you become antibiotic resistant, well, now you have a superbug and it's going to kill everybody. And so phenols were actually, well, at least triclosan was inducing non-specific efflux pumps. And so you would expose a bunch of bacteria like in a hospital to phenols, and then all of a sudden all these bacteria develop the ability to be resistant to antibiotics. Yeah, that's not good. If you've worked in a hospital setting, you may have used quat before, quaternary ammonium. So those are just a variety of compounds, but basically you have a nitrogen with 4 bonds to something else. So in this case, it could be in a ring structure. In this case, with benzalkonium, you've got your nitrogen bound to 4 different carbons, and then there's other things after that. A lot of them will have these nice hydrophobic tails, which allows them to fit into plasma membranes because it looks kind of like a phospholipid bilayer, and it just fits in there and can disrupt the membrane. A lot of these in this context would be considered cationic detergents. So cations are positively charged, anions are negatively charged. So this nitrogen has a permanent positive charge, so it's a cation. So we would call this a cationic detergent. Well, what's a detergent? Well, that's a good question. So detergents and soaps and surfactants, those are things that look like phospholipids, and so they can disrupt phospholipid bilayers. In order to visualize how this is working, a really nice video that got made recently because of coronavirus was this video that talks about how coronavirus can be disrupted with soaps because it's destroying that plasma membrane.
Washing your hands with soap is possibly the simplest way to flush pathogens down the drain. Soap molecules can interact with pathogens, such as coronavirus, leading to their destruction. Each soap molecule has 2 parts. The head is hydrophilic, which means it likes to interact with water, while the lipid tail is hydrophobic, which means it wants to get away from water to find similar water-avoiding particles. This is the basis of how soap works. When soap molecules encounter lipid particles on our skin, the tails aggregate around them to form spherical structures called micelles, creating a water-free environment. Similar things happen when they encounter enveloped viruses such as coronavirus. Envelope viruses carry their genome and supporting proteins inside a lipid bilayer membrane, with the proteins necessary for infection embedded in the membrane. In cells and viruses, These lipids are packed neatly into 2 sheets with the lipid tails facing inward. The lipid ends of the soap molecules are attracted to the lipids in the membrane. They take advantage of the presence of membrane proteins, which can perturb the neatly organized bilayer, to insert themselves into the viral membrane. If there is only a small amount of soap, the soap molecules only loosen the membrane. But with more soap, they begin to create micelles around membrane lipids. They are also attracted to hydrophobic amino acids in the membrane proteins, extracting them from the membrane. The structural integrity of a virus is essential for infection. So, washing your hands with soap protects you better from infection than washing with only water, and the longer you lather, the higher your chance of destroying the virus.
Something important to just think about, because we haven't really talked about viruses yet in this class, is that viruses steal their phospholipid bilayers from their host cell, and so they don't make it themselves. So if you disrupt it, the virus has no way of fixing that problem, and so soaps can just really wipe out a virus really fast because there's no way for it to fix it. So wrapping things up, I wanted to show this chart that just covers some of the things I talked about. It covers some of the things I didn't talk about, because there are tons of different chemicals that you can talk about, but hey, you can only do so much. We'll see another similar chart with antibiotics next week. Something else that we'll also see next week is disc diffusion. So if you wanted to study, how effective an antibiotic is at killing something, then you might do something with like a little paper disc, and you soak that paper disc in whatever you're trying to use. So in this case, you got like bleach, you got your quat down here, you got a phenol, and then you put it on a plate covered in bacteria, and you see how far the bacteria actually get. Can they actually grow near the bleach, or we would call this a zone of inhibition right there. And so how big this actual line of bacterial growth, so there's bacteria growing here and bacteria not growing here, how big that zone of inhibition is tells you how susceptible that specific bacteria is to that chemical. And we're going to see this. This would be a great way to study antibiotics as well. The very last slide I have is talking about mean inhibitory concentration, which we're going to talk more about with antibiotics, but basically what concentration of disinfectant do I need to use to make sure that I'm actually inhibiting as much bacteria as I need to. And so you can see here, we would have a few different tubes of bacteria, and you'd say, okay, replace the antibiotic with whatever chemical, like quat or whatever. And so you say, okay, I don't have any quat in there, and all the bacteria are growing. Okay, awesome. I add 1 microgram per mil, and it's starting to get a little less cloudy, and the bacteria are dying, and then I double it, and I have even more, and then I triple it, and then I've actually completely eliminated whatever bacterial growth was there. Well, that's my MIC, or your mean average inhibitory concentration. So in this case, I would say, okay, well, how much did I have to actually use to inhibit that bacteria? Well, I had to use 3 micrograms per mil, or whatever it is. And we're going to see that more with antibiotics as well.